Hello everyone, welcome to my channel, and today I am going to discuss about plant cell contents. So plant cell consist of various contents which are either food storage products or the waste products of plant cell metabolism or the byproduct. So they are identified with the help of microscopical and physical examination. There are various cell contents which are pharmaceutically important. They are starch. Now starch is present in various parts of the plant such as seed. They are basically carbohydrate, fruit, root, rhizome, and even in the chlorophyll of the leaf. So they are of various shapes and sizes and are differentiated on the basis of size, shape, position of hilum, etc. Example include potato starch, maize starch, etc. Now these are chemically polysaccharide containing beta amylose and amylopectin. Starch is basically identified by iodine test, which turns starch blue to violet. So this is potato starch. The hilum is accent in the center. In maize starch, it is not in the center. The hilum is not in the center. So this is the difference between the two starches, that is potato starch and maize starch. Next is eleuron grain. So protein is stored as eleuron grain in plants. The mass of protein is enclosed by a thin membrane in eleuron grains. It occurs in the endosperm of the seed. The protein mass often encloses an angular body known as crystalloid, or rounded bodies known as globoids. Now deflated thin section of eleuron grain, when treated with alcoholic picric acid, the crystalloid and brown tissue stains yellow. Next is fixed oils and fats. Fixed oils and fats. Now they are found abundantly in the vegetative as well as in the reproductive parts of the plant. Fixed oil is concentrated as reserve lipids in the seeds. Fixed oil occurs as refractive oil globules. They occur as in association with eleuron grains. Test is 1% osmic acid solution colors them brown to black, or either brown or black. Next is calcium oxalate crystals. Now these are excretory product of plant metabolism, and they are of various forms and diagnostic of crude drugs. Example include microsphenol or sandy crystals, belladonna. Now they are tested by clearing with caustic alkali solution, because caustic alkali dissolves the crystals. Now we test them under the polarized microscope. So first of all we take the cleared sample, and then we put some reagents like caustic alkali, in which it is insoluble, and if we put 60% weight by weight sulfuric acid on standing, it replaced by calcium sulfate crystals. Next is calcium carbonate. They are also known as cystoliths, and they appear as small bunches of grapes in the tissue. Now they produce effervescence when dissolved or when put in hydrochloric, sulfuric acid, etc. And if we put them in 60% weight by weight sulfuric acid, needle shaped crystals of calcium oxalate separates out. Mucilase, these are polysaccharide complexes of sugar and uronic acid formed from the cell wall. They are insoluble in alcohol but dissolve or swell in water. Their identification is done by ruthenium red, which stains them into pink color. So this is a brief discussion regarding the different cell contents present in the plant, which are of very necessary or important pharmaceutically. And here I am ending my today's discussion. See you in my next video, and if you guys like my videos, please subscribe the channel. Thanks for watching.